Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Building Efficiency into Biologics Formulations Development. My name is Edward and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive and webinars work best when you're involved, so please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for future streaming on xtalks.com. At this point, it's my pleasure to thank Curia, who developed the content for today's presentation. Curia is a contract development and manufacturing organization with over 30 years of experience, an integrated network of 29 global sites, and over 3,500 employees partnering with customers to make treatments broadly accessible to patients. Their biologics and small molecule offering spans discovery through commercialization with integrated regulatory and analytical capabilities. Their scientific and process experts and state-of-the-art facilities deliver best-in-class experience across drug substance and drug product manufacturing. From curiosity to cure, they deliver every step to accelerate and sustain life-changing therapeutics. And now it's my pleasure to introduce our speaker for today's event. Dr. Tyler Jones is the Director of Formulation at Curia, focused on formulation and cGMP fill finish of injectable biologics. Dr. Jones has over a decade of experience in the biopharmaceutical industry with previous employment at companies including Nectar Therapeutics, Avanti Polar Lipids, and Integrity Bio. His expertise is in the field of biologics formulation development, and his PhD training was focused on protein biochemistry, structure function, and biophysics. While at Curia, Dr. Jones has worked on development projects for over 80 unique molecules, including proteins, peptides, nucleotides, lipid nanoparticles, and more. He holds a PhD from the University of Alabama in Huntsville. And now, without further ado, it is my pleasure to hand the mic and presentation over to our speaker, Dr. Tyler Jones.
Thanks, Edward, for that great introduction. I appreciate X-Talks hosting this webinar, and hopefully we'll all have some fun and bounce some good formulation ideas around. As Edward said, my name is Tyler Jones. I'm the Director of Formulation at Curia's Camarillo and Thousand Oaks, California sites. Here at these sites, we specialize in drug product development, clinical fill finish. For the past many years, I've been working in biologics formulations development. I learned everything I possibly could from Dr. Byung Chang and others. These are mentors, people that should be on the Mount Rushmore of biologics formulation development, if that was a thing. It should be. I'm sure it would be well-attended attraction. But today I really want to focus on and think about how we can do a really good job as formulation scientists, but also play nicely with the rest of the program, perhaps acquiesce to some of the limitations that are inevitably going to be placed on our work. A better way to perhaps put this is, if we had unlimited funds and unlimited time, we could always make incredible formulations within the laws of physics, right? But we will never have unlimited funds or unlimited time. So we really have to be wise about what we do with that. That's really the framework of what this talk is about. So let's jump in. Edward already covered my bio, so I can skip this. I don't really like talking about myself anyway, so that's great. But this does have my contact info there at the bottom for posterity. Please feel free to contact me with any questions or just to talk about pharmaceutical development, formulation, Alabama football, music. It really doesn't matter to me. Feel free to reach out anytime. So for a quick overview of what we plan to cover today, the first section will be a general overview of formulation development for biologics. We'll talk about goals, where we fit into the overall product development and life cycle. And then we'll get more specific with different approaches to formulation development. We'll talk about some different tools and tricks that we can use to build efficiency, but also the pros and cons of these various approaches. This will be very important because unfortunately, most of the time, you can use some tool or approach that will save time and money, but also you usually get what you pay for, right? So there are cons that should be well understood before going down some of these paths. It's really overall to look at different approaches, speed versus being very thorough, and also cost versus an assumption of increased or decreased risk. That's really the name of the game here. I'll try to get through this overview quickly, but I do think it is kind of important to frame the conversation that we're going to have. So we all have this ultimate goal of creating a well-characterized biological product. That term came directly from the FDA in the 90s when biopharmaceuticals were really taking off. We also have some more lofty goals tied to some target product profile that is usually given to us. As a formulation scientist, I can say that I would love it if every single target product profile was low concentration, frozen formulation, so on. My job would be very simple, but obviously this isn't commercially viable, so we do have to work within some bounds. So to achieve this ultimate goal, we have to develop a deep understanding of the behavior of the molecule of interest, the critical quality attributes of the product, and how these attributes are also affected by all aspects of the process and not just an ideal closed system. Identifying and understanding all of the critical quality attributes of a product is difficult enough for a well-behaved molecule. Now add in this layer of complexity that you have a very fragile and unique macromolecule that is susceptible to all sorts of stresses and degradation. And it really begins to take a large team of people with great communication to solve all of these problems and produce that well-characterized product. Ultimately, this leads us to the quality by design approach, where a deep understanding of those critical quality attributes I mentioned is developed. And as you'll see, these attributes can be and actually are connected to each other, to upstream and downstream manufacturing processes, or are even direct results of manufacturing itself. Critical quality attributes are any attributes that have any impact on the efficacy or safety of the product. So from a broad perspective, we as formulation scientists need to have that understanding that all of these issues eventually do have to be worked out before you get to that quote, well-characterized biological product. And even things like the manufacturing process, which we're not usually directly involved in, but probably should be, plays a huge role in the final product itself. So as an example, here I've listed some critical quality attributes of protein-based pharmaceuticals. As you can see on the left, we have product-related impurities. This is where we spend a lot of our formulation efforts trying to mitigate issues with aggregation, subvisible particles, chemical changes like deamidation, oxidation, or disulfide shuffling. These are all major common hurdles of protein formulation development. They are going to be product specific, and different molecules will have different issues, but in general, these are quite common, and formulation scientists have tools to attack these issues. On the right side, we have process-related impurities. Here we begin to see the upstream and downstream manufacturing considerations. Upstream, you have potential carryover from any of the upstream processes. Hopefully, a well-developed drug substance supply will take care of these issues for you, but it's good to know that it's there. On the manufacturing front, the fill finish of drug product in particular you also have considerations that affect the final performance or stability of the formulation. In an ideal world, right, the exact components of what you say you want to put into the vial are all that go in. So for example, let's say I have this formulation. It's 20 millimolar histidine, 7.5% sucrose, 0.02% polysorbate 80, and that's at pH 7.2. That's the idea, but unfortunately, there are extractables and leachables, raw material related impurities. The polysorbate or the histidine or the sucrose, it's not going to be 100% pure. They should be close if you're using, good compendial GMP ingredients, but not always 100% pure. So not all of the critical quality attributes are related to the exact components you're trying to put into the vial, or even related to the intrinsic degradation species of your API, but they're also connected in various ways to each other and to upstream and downstream processes as well. As an example of this connectivity, we've worked on a lot of proteins in the past that are susceptible to oxidation. It's very common. Most people are familiar with it. We may be able, in our R&D lab here in Thousand Oaks, at small scale, we might be able to produce a formulation through dialysis and then manual filling that shows very little oxidation. But when you take that and you run it through a manufacturing process, we might introduce metal contaminants from contact surfaces or pumping through filling needles. And these metals can then catalyze oxidation, even at very low concentrations, right? Parts per million, they're still there and they're still catalyzing oxidation reactions. And so this is a real example of formulation development process that we've gone through in years past. We were able to actually identify and understand that potential downstream influence on the formulation through robustness studies, and then we could add chelators or antioxidants, which proved to be a crucial part of the formulation itself. So overall, successful formulations are safe to use. First and foremost, safety and efficacy. So safe to use, effectively deliver the drug to the target. They're stable during manufacturing, storage, and temporary exposure to unexpected stresses. I'll touch on that a bit later in the approaches section. Good formulations also, they have to be compatible with the delivery route. They have to be compatible with devices, methods that you use, and as always, market competitiveness is important. IP issues are something that have to be taken into account. So there's a lot to think about when approaching formulation, and it's fun. It's a good challenge. It's like a huge puzzle, and it's very enjoyable. So now we kind of have a 10,000-foot view of the goals and the complexity of biologics formulations development. So how do we actually go about tackling this challenge? Well, it's a difficult question, but there's probably about as many different ways to approach formulation as there are formulation scientists out there. Everyone will likely have their preferred processes and approaches, and I'm not here to say that any one approach is the definitive correct approach. That's not the purpose of this talk. I just want to share knowledge and look at this from a step back, right? And to do that, you really want to put yourself in the mind of a product lead, head of CMC, a vice president. These are the decision makers that direct where to spend effort and how to spend money wisely. How much time do we have? These are all things that they have to wrestle with, and you really want to look at this problem from their point of view as well. Oftentimes, these are the same people that are also looking for some guidance and expertise. They might not be formulation experts, right? And they do appreciate when a formulation team can understand their needs and be flexible and not just give them a menu of package options, right? So with that, we have to think about phase appropriateness. What is phase appropriate for what we're planning? Is it wise to spend 12 months developing some deep NMR structure binding dynamic experimental data on some new drug substance before you actually get into drug product formulation development? No, I mean, in my opinion, no, not at all. That's kind of a goofy thing to do, especially when you can pragmatically get into an advanced formulation much quicker and start developing long-term stability data with more efficient approaches, right? So science and business need to be balanced for efficiency without losing effectiveness. Let that sink in for a minute. So science and business need to be balanced for efficiency without losing effectiveness. That is the crux of what we're talking about today. In this timeline, we see the various stages of product development: discovery, preclinical, process development, drug substance development, on through IND, clinical trials, and then eventually on through market, right? In its simplest form, you can think of two major formulation phases, although there can be more. First and foremost, at the early stage, you want to ask yourself and also answer the question, what is phase appropriate? Most of the time, trying to get to this step, the phase 1, 2 clinical trials, the first in human, people are in a huge hurry. Most molecules don't make it past that. So spending years and years doing research only to fail right there it's really deflating. You know, it's not a waste, but it feels like that. So almost everyone just wants to get into tox studies and get into first in human as fast as possible. Does the formulation need to be perfect for that phase? The simple answer is no. A good enough formulation to support clinical trials is fine. I say that with air quotes, good enough formulation. Maybe that looks like 6 months of stability in a refrigerator or a frozen state. Maybe it's a lyophilized formulation. This is all acceptable, and it will keep the program moving forward. When the molecule gets into clinical trials, you've then bought yourself a lot more time to continue working on the formulation, optimizing it. You don't want to be the roadblock of progress in formulation. Let other people be that roadblock. So tips like this are great, right? Get a quick and dirty formulation done, get out of the way, and keep improving the formulation concurrently with other actual roadblock activities. I'll talk a bit more on how to approach that first good enough, quick and dirty formulation in the next section. But for now, I want to look at the formulation flow itself. So this cycle shows the large variety of types of formulation research that we actually go through. These are not exactly in a perfect order for every single project. There are some things we want to consider and think about at various points of development. For example, are you developing a multi-dose formulation? Do you want to wait until after you've gotten pretty far along in stabilizing your molecule through normal formulation practices before you start to look at preservatives and things like APET, antimicrobial preservative effectiveness test? No, absolutely not. If possible, try to check on that as early as possible in the process, as you can feasibly. You don't want to find out that your molecule doesn't play well with a lot of preservatives, maybe only works with a specific combination of preservatives, and then the pH of your developed formulation is outside of the activity range of that preservative. That's just an example of taking the appropriate time in the design phase to think ahead. So this is the design phase, right? Think ahead, have forethought about eventualities. This is presented as a cycle because formulation work is never really done. Even after products launch onto markets, they're almost always continuing to work on product lifecycle support. changing to a more competitive presentation, for example, intellectual properties. Things like that will add lifetime and value to the products, and they all involve formulation support. So the job is never really done here. Over the next few slides, we're going to think about a few concepts that are critical when thinking of science and business being balanced for efficiency without losing effectiveness. I put together this silly little infographic showing the classic tortoise and hare concept. You can think of opposing sides of a seesaw being speed versus thoroughness, and also cost versus risk. These are four things that influence each other. For example, the more thorough your research is, the less risk you assume in your decision making. Generally, the quicker you do things, it might be less expensive, but it can also increase your risk at the expense of being less thorough. So with this tortoise and man-shaped hare race that we have here, it may take longer and it might be more expensive to do something, but it also increases your chance of success. Sorry. So yeah, here the tortoise has a better chance of increasing his winnings at the finish line because he has the better chance of actually getting there. The faster, less expensive option has a greater chance of stumbling along the way. And this is the balance and framework that should be in the back of our minds when we think about different ways to approach formulation, generate data, make decisions based on that data, and just move forward. Earlier, I said it would be great to have unlimited time and unlimited funding to do formulation work, but let's be honest, that's not reality. So we have to strike a balance, and we have to understand the implications of what we do and the decisions that we make along the way. I also mentioned earlier, put yourself in the mind of a product lead, CEO, a CSO, vice president, head of CMC, these people, the decision makers that direct where to spend effort, money, and how to drive timelines. They have very important decisions to make, but they themselves are also beholden to budgets and investors. Everyone on this call is most likely aware of how the biopharmaceutical industry operates. There are a lot of companies that are startups, virtual companies, and these companies and these people have incredible ideas to cure all kinds of diseases with cool new technologies and new drugs. A lot of the time they're running on Series A funding, and they need to get proof of concept as fast as possible. So that leads to the first strategy here I like to call quick and dirty formulation work. This is so common. It may be a case of discovery has 4 or 5 candidates that need to be whittled down, or let's get this into first in human as fast as possible. Many times with this approach, you see things like platform formulations. These are most likely to succeed type formulations. They're usually somewhat finalized looking formulations with a lot of components, and are often based on precedents, what has worked in the past on this type of compound that you're working with. For example, let's say I have an IgG2, the most common therapeutic monoclonal antibody out there. People that work in protein formulation development have very likely done more IgG2 work than anything else. And luckily, these antibodies are usually quite well behaved. And I say usually because that doesn't mean they're always well behaved. So with this approach, we can test 4, 5, 10 different formulations from, say, pH 5.5 to 7, some sugar, maybe some salt. There's usually a surfactant, polysorbate. There's a decent chance that you could find a stable enough formulation to test stability. You can run very short screenings at accelerated temperatures. You often see things like DSF or DSC screening, differential scanning calorimetry. This is used to whittle down pH space, and then subsequently again to whittle down sugar selection, concentrations, things like that. And these are basically instantaneous readouts, right? This is very quick experiments, going through an entire battery of different conditions might only take a few days max. So going back to our speed versus thoroughness and cost versus risk metrics, this is very quick. It's usually quite cost effective. However, it's definitely not thorough. And as I mentioned, you are therefore increasing risk. For example, let's say you make a formulation decision, or at least significantly narrow your formulation space down that you're looking at, based solely on some instantaneous DSF data, thermal unfolding. And then 9 months later, when you have actual real-time stability samples set out, you start seeing problems. You know, what's happening? You don't have a lot of knowledge base to explain this or investigate what may be happening in your formulation. And this happens way too often. So how can we increase our base knowledge and reduce that risk? So that leads to my second concept, which is approaching formulation with a more pragmatic, relevant screening process. Think about simplifying variables so you can understand what each variable is doing. For most modalities, the base variables that really drive how the molecule behaves are pH and ionic strength. For proteins, surfactant is usually quite important as well. So you will want to screen surfactant as early as possible. If you do a lot of other screening without surfactant or with the wrong surfactant, you may very well be seeing just surface active related issues, and your other variables such as pH or ionic strength are being masked. The degradations you see might simply be able to fix with the correct surfactant present. And surfactant can be a bit more agnostic to the rest of the formulation components than vice versa. The other key piece of this approach is the screening itself, what stresses and methods to use. Really, in about 8 weeks of real-time stability across a range of temperatures, you can differentiate formulation space quite well. It's critical to look at a range of temperatures because many protein degradations are non-areneous in behavior. That means something that you see happening at 40 degrees Celsius may never happen at 5C. However, at 5C, you may not see a large difference between formulation at pH 5 versus a formulation at pH 6. So collecting the accelerated data at higher temperature is also important to force differentiation. This is all about taking into account the possible degradation profiles, but also understanding which ones are more relevant to your intended storage temperature. So there's a lot of weighting and factors that have to be looked at when making formulation decisions with this approach. So in contrast to the quick and dirty approach, this approach is trying to develop that deep base understanding of the molecule's behavior. If you look at a broad range of pH, and at each pH interval you look at both high and low ionic strength, that vastly different electrochemical, physicochemical environment that you've created will force the protein down different degradation pathways and different journeys. This allows you to understand and identify the critical quality attributes of degradation products because you will likely be creating most of the possible ones across such a broad space. However, you also have the ability to hone in on a formulation sweet spot. You then have a robust data set to base any further formulation decisions on. That's the key. For example, let's say at pH 5 with low ionic strength, you see the best physical stability and deamidation rate is low. However, you still see oxidation occurring. Well, that's great. That's a simple fix. you can then screen some antioxidants and pointedly mitigate that issue. And you have the base data to show the FDA why you chose to add that extra excipient to your formulation, which is in contrast to a platform formulation that just starts with everything already there. This is sort of building a formulation from the ground up. And I said it before, isolating the variables so that you understand what each variable is responsible for. The central piece of this approach is being thorough, building out a robust data set to base decisions on, not making assumptions or using historical precedents from other molecules and hoping that it works for this new one. So looking back at our speed, thoroughness, cost, risk metric, this approach may not be as fast as a quick and dirty approach, but it significantly de-risks your decisions. Overall, in the grand scheme of a product development, this 8 to 12 weeks total to build out this robust data set is a drop in the bucket, honestly. This work can be done on engineering run material from drug substance scale-up work. So it definitely doesn't need to be a rate limiting factor at all. Our next approach isn't really an approach to formulation. It's just something that has to be done. Eventually, you will have to put samples down on real-time stability. This follows ICH stability protocol to help prove stability and set expiration dates. This just takes a long time, honestly. However, if you look at this as an opportunity instead of, oh no, I have to do a long-term stability study, you can use this opportunity to further refine a formulation. A lot of people use this phase to look at a variety of things, fine-tuning conditions like small pH steps, small adjustments in buffer strength, or excipient percentages. These can be done with a DOE approach, which is great. Some may opt to prepare these formulations in a manner similar to manufacturing. So process challenge, right? Looking at UFDF, put it through all of the process challenges, pumping, filtration. We've even, here in my lab, we've even filled long-term stability samples through filling needles and pumps. As I mentioned earlier, oxidation potential from these new contact surfaces. It's a great opportunity to close any loops, answer any final questions you may have about formulation, and there's an endless amount of activities you can incorporate here. You just want to be very careful and thoughtful about how you embark on this journey, because it is a long journey usually. So, forethought, good planning, just be extremely careful before you start one of these long-term studies, because it takes forever. So for my last number of slides, I want to look at some pros and cons, and then also talk about some pitfalls and things that you want to be wary of. Just quickly reviewing these different approaches. If you are in a pinch and up against the deadline, or you have a small budget, you can perform very fast formulation work and potentially succeed. But there is a larger chance of failure when making decisions on smaller data sets or data built on perhaps not as relevant conditions as real-time stability. I'll discuss this a bit more in a minute. The thing with the quick and dirty approach, it's more of a gamble. You can get to a formulation quicker and cheaper, and it can work. Eventually, even through this path, eventually you can perform all of the supportive studies on that formulation to round out the knowledge gaps that are missing. Force degradation studies can prove your stability indicating assays are working. All of the other robustness and anything else you need to go in a filing. It's just a question of whether you are comfortable or have the ability to risk that the smaller formulation work will succeed. If it does not, you end up doing even more work than you would have initially if you were just to start with a more thorough approach to begin with. If you take the critical quality attribute identification, formulation space characterization route, you will de-risk your decisions, and you'll have more relevant data and a better understanding of formulation variables, but it is not as fast, and it may cost a bit more than a quick platform, just very short approach. But again, is 8 to 12 weeks in your 12-month timeline a showstopper? Not at all. Not if you plan ahead and you get onto formulation early. You'll almost certainly still be waiting on that final GMP drug substance run well after your first drug product formulation is ready to go into drug product GMP fill finish runs. And lastly, here we have the good old long-term stability study. Again, just has to be done at some point. It's a good opportunity to refine formulations, but obviously takes a long time and costs a good bit of money. It's usually quite a long study. So I've mentioned relevancy, I don't know, 50 times so far. I really put emphasis on this term because I've seen it too many times. Honestly, I don't know how many clients I've worked with that have had formulation work done quick and dirty, and the majority of the decisions were made using thermal analysis data. And then 6 months into a long-term stability study, they start to see issues with their formulation. There is a good reason for this. If you think about it, if you look at the activation energy of thermal unfolding of proteins, which generally happens at 50, 60, 70 degrees Celsius, higher temperatures, and then you try to extrapolate that down to 5 degrees C, it's literally hundreds of years for that to happen in a refrigerator. Proteins sitting in a refrigerator are not undergoing thermal unfolding. You're storing your drug product in a refrigerator. So why is this used to eliminate so much formulation space so quickly? You have to be cognizant of what degradations are relevant and may actually occur based on your intended use or intended storage. I'm not saying that this can't work. I'm just saying be careful putting heavy weight on this type of data to make important decisions. This is a quick reminder that analytical methods are your friend. What tools you use to analyze formulations and compare and contrast results are the most important piece of all of this. If you have a platform size exclusion method and screen a limited set of formulations and you don't see much happening, no aggregation formation, for example, you may think that your protein is just very stable and move on, or your method may not be separating well and you do actually have aggregate that you just can't detect. This has happened. This is a cautionary tale. It didn't happen to me, thank goodness. A company I will not name, it's pretty large, got a protein into clinical trials and first in human, and they started seeing some adverse reactions. And it turned out someone had developed a size exclusion method that had, I think, methanol or something in the mobile phase, whatever it was an organic in the mobile phase that was dissociating the aggregate, so they weren't detecting it on the size exclusion method. I mean, almost everyone knows not to do that, but somehow that actually happened. And it's just it's a cautionary tale to be vigilant about your analytical methods. Make sure that they are stability indicating and work with the formulations you're using. If you develop a new ion exchange HPLC method, for example, do your formulations that you're looking at have high or low salt? What's the injection volume? If you inject a high salt formulation at a higher injection volume on an ion exchange column, it's going to affect the performance. So the outcome of the method depends on you having a knowledge of all of these possibilities and considerations. And pointed out here just so that everyone is acutely aware that analytical methods are what we base all of our decisions on, and we really, really want to make sure that they are performing well and giving us relevant real data. Just like any science, you want to make sure you have the appropriate controls in place, positive and negative controls, to make sure that what you're seeing is real. Lastly, I do want to mention this. I don't have time to go into too much detail. At the end of the day, whatever formulation you produce will have to go through manufacturing, which comes with its own set of stresses that proteins are susceptible to. I went over the importance of this early on in the first section. I just really want to hammer down this point. It is wise to include these types of stresses alongside temperature stability stresses, so that you don't get too far down the road with a formulation only to find out that it can't withstand a freeze-thaw cycle or falls apart when circulated through a TFF system. In the same vein, you have other robustness testing that it isn't manufacturing related, but things like in-use container closure compatibilities. There's a massive amount of work to be done around verifying that every single contact surface the formulation will ever see won't adversely affect that safety and efficacy. Just for example, silicon tungsten compatibility studies for products in pre-filled syringes. The plungers of the PFS are often lubricated with silicone oil, which may or may not interact with a protein. They're known to cause aggregation, things like that. Heated tungsten pens are used to mold the channel where the needle is mounted. This creates tungsten oxide vapor that can deposit in the syringe funnel. And these are just single examples of the, so many different things that a formulation has to withstand. With that, if you're a good formulation scientist and you're listening to this, good luck, my friends. Keep fighting the good fight. I do want to thank X Talks again for hosting this, and I especially want to thank my team at Curia, Camarillo, Thousand Oaks. I don't have enough space to list everyone here, but they are all rock stars. I'll be happy to answer any questions, so I'll go ahead and pass it back to Edward to moderate the Q&A. Thank you again.
All right, and thank you, Dr. Jones, for that very insightful presentation and for all the shout outs there. We very much appreciate it. As Dr. Jones mentioned, I would like to invite our audience to continue sending in their questions or comments using the questions window for the Q&A portion of this webinar. We have already received some questions, so let's get started with those. The first one here for you, Dr. Jones, is viewer asking, a lot of the focus was on proteins, but do these approaches hold true for other biologics like LNP, peptides, vaccines?
Yeah, that's a great question, actually. In short, yes. All of these ideas work quite well for other modalities. You can use best guess platform formulations based on what's already out there for lipid nanoparticles or vaccines. But developing that critical quality attribute profile and gaining an understanding of what the drug is susceptible to is quite important for these other modalities. Some may be highly susceptible to freeze thaw, or we've seen dehydration upon lyophilization being an issue for some things. Peptides often come with their own slew of issues, just the nature of them. They can often be highly pH sensitive in terms of solubility and ionic strength. They seem to be, like easier to salt in or salt out, but because they're so small and not as structured as proteins, which proteins typically have buried hydrophobic cores and patchy surfaces with various charges, peptides are mostly solvent exposed, so every amino acid comes into play. There are pluses and minuses to this. A lot of peptides are more stable and don't degrade as easily, but also other peptides can be much more difficult. It really just depends on the sequence and the peptide itself. But in short, just to answer the question, yes, these approaches hold true for basically all biologics modalities.
I see. Thank you very much for that answer and for going in depth with all those other biologics. Another question for you here. This viewer asking, what types of issues may occur over longer term stability in formulations that were developed with a quick and dirty or platform method that weren't able to be picked up with earlier screening?
Yeah, that's a great question. I can tell a story that I saw personally around this idea. We've had a project in the past come to us that was they were at about 12 months of stability on a long-term stability program, and they had gone through that quick and dirty screen. When presented with their issue, and after asking to be, taken through the formulation development history up to that point, they really all they had was some thermal analysis data to show why they chose the formulation they had. And they started seeing around that 9 to 12 month mark, they started seeing accelerated aggregation subvisible particle formation well into the study. So unfortunately, without much base characterization knowledge, it was quite difficult for us to say just like, hey, well, here's your issue right here. We just don't have a lot of data to make that kind of call. So it ended up being a drawn out process where we more or less restarted formulation work trying to better characterize the behavior of the protein. We did do some particle identification and found the particles were a mixture of proteinaceous particles mixed in with some degradation species of polysorbate. They had used a lower grade of polysorbate that can basically hydrolyze itself by some of the impurities. It's a long story, but in the end, they weren't in the ideal formulation space, and it did end up costing them a lot of time, unfortunately. But that's why I bring up these issues, because I mentioned earlier I've seen it many times at this point.
I see. Yeah, definitely a tough position to be in there. But thank you for sharing that story. Much appreciated. I have another one for you here. This audience member asking, what if we don't have an established target product profile? How would one know where to start with formulation development?
Yeah, that's a good one. This happens quite often, honestly, especially programs that are just getting kicked off. So this goes back to oftentimes to that get as fast as possible into first in human, or maybe looking at multiple candidates from discovery. Ultimately, my advice is, if you have the time and ability, try to do some thorough work to understand what you're getting into. If you have absolutely no idea what concentration you want to be at, just start with something reasonable. You can base it on what you're getting out of drug substance production. Are there, huge issues with solubility in drug substance production? I may have to start at a very low concentration to find a more suitable pH versus ionic strength space to be in. And once you do that, that knowledge can then be pushed back upstream to help improve drug substance purification. We've seen products, more than a couple, that have had really terrible issues. We've been sent inclusion bodies, and clients have asked to see, what can we do with them. So even if you have, no guidance or knowledge or any idea what kind of target product profile to go for at a very early stage, formulation groups can still help. They can help do some scouting work to guide some of those very early decisions.
All right. Well, thank you very much for those tips and also for that very simple yet valuable answer. Greatly appreciated. At this point, we have in fact reached the end of the question and answer portion for the webinar. So thank you again, Dr. Jones, for all of those answers. If we couldn't attend to your questions, the team at Curia will follow up with you. Additionally, as Dr. Jones mentioned earlier, if you have further questions or you want to chat sports or whatever, you can reach out at the email address showing on your screen. That's tyler.jones@curiaglobal.com. Again, thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve our webinars. I'm also going to send everybody a link in their chat box. You'll be able to view the recording of this event via that link. And additionally, if you'd like to share it with your friends and colleagues, XTalks and Curia would greatly appreciate that. Once you share this link, other friends and colleagues will have access to the recorded video for today's webinar. Again, I'd like everybody to join us once more in thanking our speaker, Dr. Tyler Jones. Dr. Jones, thank you again for a really insightful and very well put webinar.
Yeah, thank you very much. I appreciate you having me.
It's our pleasure. It's our pleasure. We hope you all found this webinar informative. Have a great day, everyone.